There's a dog named Cliff in the Netherlands who's been trained to detect the chemical signal, volatile organic compounds, off of Clostridium difficile, which is a very serious life-threatening infection, mainly associated with hospitals. He can detect it in individual patients and hospital outbreaks 3 days before any instrumentation or other technology. And think about how dogs greet each other. Is it face-to-face? So I contend they're actually asking, who are you microbially? How healthy are you? And might you be willing to share? In many cases. I used to yell at my dog, say, don't do that. And now I'm like, you guys are smarter than I thought.
On behalf of Harvard Bookstore, I'm pleased to introduce Rodney Dietert discussing his book, The Human Superorganism, How the Microbiome is Revolutionizing the Pursuit of a Healthy Life. Rodney Dietert is a professor of immunotoxicology at Cornell University, where he has also served as director of the graduate field of immunology, the program on breast cancer and environmental risk factors, and the Institute for Comparative and Environmental Toxicology.
It's such an honor to be able to have an event here at the Harvard Bookstore. I really appreciate it. When I think about all of the authors that have been here over the years, brilliant authors, all of their works, carrying their microbes with them, their microbial bubble, and maybe even leaving a few behind, so I hope this hasn't been sanitized too much, actually. But I really appreciate this. So I was hired into Cornell way back in 1977, trained in immunogenetics, and I was actually supposed to make healthier chickens by improving their natural immune system so that they could resist disease better naturally. There wasn't too much money for genetics on higher vertebrates at that time for research, so I wound up immigrating by research funding opportunity into environmental health and into toxicology, and basically was self-trained in those at Cornell over enough decades. So that's what I worked on, and the consistency of my research has been on protecting the developing immune system, first in baby chicks and then in children, baby humans, and trying to prevent disease. And in particular, what we now call non-communicable diseases, what they used to be known as chronic diseases. And that's really, here at the latter stage of my career, my goal is to do something useful to blunt what is really an out-of-control epidemic of non-communicable diseases, or what we call NCDs. So the book, The Human Superorganism, is advertised, and appropriately, that it's about the microbiome, because it is. That, I believe, is the solution to the epidemic. But in fact, the book is really about this out-of-control epidemic of diseases that are the 21st century diseases versus the earliest 20th century diseases. And what we've been doing to date simply has not been working. As well-intentioned as smoking cessation, dietary changes, and the like, it hasn't blunted those diseases. And so that's my goal. And I'm really hoping that this book, through both encouraging self-help in terms of wellness, but also a different healthcare and medical approach to the human, a different way to think about humans, is really going to be useful in terms of these diseases. So I'd like to start, if you'll humor me, with a little exercise. And that is for me to read off as many non-communicable diseases as I could get on the sheet of paper. And if I'm showing slides, it's on one slide. And simply ask about your circle, like your network, your telephone, cell phone network, your circle of friends. So this would be family members, those you socialize with, co-workers, co-students, anybody you're connected with. If they have received any one of these diagnoses, as you hear it, if you just raise your hand. Allergies, food allergy, asthma, rhinite, allergic rhinitis, hay fever, dermatitis, cancer, any form, obesity. You can keep your hands up, so if you don't mind, it won't be long, we'll be done. Type 1, type 2 diabetes, cardiovascular disease or heart disease, arthritis, autism spectrum disorder, ADD, ADHD, celiac, inflammatory bowel, so Crohn's and ulcerative colitis, lupus, autoimmune thyroiditis, so Graves and Hashimoto's, depression, osteoporosis, frailty, dementia, Alzheimer's disease, hypertension, sleep disorders, PCOS. That is an ovarian chronic condition. COPD, the chronic obstructive airway disease, chronic kidney disease, psoriasis, multiple sclerosis, or GERD. That's my personal one. So, is there anybody that didn't know someone with one of those? Would be, yeah, no. And that's what happens in every audience, even 500, 600. There's no exclusions. And that's because these diseases touch all of us in some way. In the last century, the early part of the last century, infectious diseases were the major killers. They were the challenge. And really, it's a remarkable story of effectiveness. Fleming, penicillin, and it's not that they've been eliminated, but when you consider cholera, typhoid, and some of those killers, the sanitation, the antibiotics have been miracle drugs. But what we didn't anticipate is what would replace them, and that's these NCDs, or non-communicable diseases. And if you look at asthma, you look at Alzheimer's, just consider peanut allergy. You didn't used to see that. And it's not something that's about diagnosis that's the problem, because anaphylaxis is pretty obvious if you have peanut-induced anaphylaxis. These didn't used to exist, certainly not in the numbers that we see. Celiac is moving to younger ages. Didn't used to really think there was pediatric celiac. There is now. All of them are moving to early ages. Even atherosclerosis, known as an adult disease, you can pick up the markers and indicators of that in childhood. Pretty good predictors that that individual is very, very likely to have that diagnosis. Beyond it, these things beget each other. So just a couple of statistics, if you don't mind. So in the book, I reference a statistic that non-communicable diseases kill 68% worldwide. So that's not just developed countries, but developing nations. While it was in advanced copy review and the like, the CDC put out a figure in February that it's 75%. So 3/4 of the world deaths are due to these diseases. So that's the target. That's the target, yes.
What's the other 25%? Is it accidents or?
And Ebola and Zika. I mean, there are infectious diseases, there are accidents, war, there are conflict. So it's a mixture of a whole variety of things. But again, giving some of those I read out is probably not, if you think about it, if you add them together, it's not shocking. The thing is, the impetus is to not add them together, it's to divide. Researchers get funded by disease. So you work on arthritis, or you work on obesity, or you work on lupus, or heart disease. And your disease is your disease, and that's your model for life, career life usually. And the federal agencies fund, and they're divided in terms of disease groups. And medical coding is by disease. And the drugs that the physician will be recommended to the physician to treat each disease is by disease. And if you get a new name disease, you may have a new drug that goes with it. So they propagate and divide. But my book is all about the benefit of looking at similarities rather than divisions. Because if you want to stop the epidemic, you don't want to just fight the epidemic of asthma, childhood asthma, and the epidemic of type 1 diabetes, and the epidemic of childhood obesity separately. They're actually more similar than different in terms of cause dysfunction. So what the book really gets at is the fact that our food has changed, but we have changed more, really within just 2 or 3 generations. And that we're not fighting the same diseases, and we haven't had a unified recognition, both for self-help and for medical care, of what to do. So the non-communicable diseases, it's not just that you die, because we're all going to die of something, right? It's how you live that's the problem. Because these diseases, I contend, and I show some examples. If I had slides, you'd be seeing what I'm going to do, what I got to do today for tomorrow's flight. Pick a seat selection. So let's say you pick seat selection and you have a diabetic adult who the Mayo Clinic has recommended that individual carry peanut products because they will help to boost sugar levels, seated in a row in front of a peanut allergic child. And you have someone celiac disease that can't do pretzels, which is an alternative to peanuts. And then you have a dog allergic child seated behind a multiple sclerosis patient with a service dog. And that happened where a child and a boy and his family were removed from a flight recently. And then you have a fragrance-sensitive individual seated next to a nicely perfumed adult. They're not compatible. We can't even get on airplanes like we used to be able to. Think about preparing a child's birthday party. What do you do for food? Think about hosting a church dinner, or hosting even a large dinner event at your place, or a science conference for anybody else that's science. What accommodations do you have to have for food that we didn't used to have to do? So it's actually isolating us. And we're running out of both health care, the Harvard School of Public Health, actually that direction I think, and the World Economic Forum have estimated that by 2030 it's going to take almost half of the world's net worth to pay for non-communicable diseases. So we can't afford it. And there is increasing stress on caregivers because autism spectrum disorder rate has gone rapidly from 1 in 150 to now 1 in 45. I had to revise the figure as I was doing the galley proofs because it changed again, got lower. And Alzheimer's, dementia, so many of these Parkinson's, so many of these diseases require assistance, and they isolate us. So we're isolated from congregating like we used to be. Many of us more and more require assistance. And when you get a drug for hypertension or a drug for heart disease, that may be for life, right? For life. So it's not that we cure enough of these diseases. Very few of them are actually cured. They're medically managed, which means drugs that you may take over decades, and non-communicable diseases beget each other. So the statistics are, if you get to be my age, you're likely, and you had one, you're likely to have more than one. And one of the things I did back in 2010 was to publish a series of papers that showed the comorbidities, what's said to be the related disease risks if you have one non-communicable disease. So we did that for childhood asthma and for thyroiditis, autoimmune thyroiditis, and for psoriasis and inflammatory bowel. And all of those you have 8 to into the teens comorbid additional diseases that group is likely to develop. Not necessarily an individual child, but the group as a whole will develop those. So with asthma, it's lung cancer, because the lungs been getting insulted for decades. With psoriasis, it's skin cancer is one of them. With celiac inflammatory bowel, it's GI tract cancers. But many, many other things as well are interconnected. And yet, when physicians treat a child with asthma, are they thinking about lung cancer at age 50? No. Now, part of it is they didn't know. But now we know. So we're not really stopping this web or network of these diseases. And what's at the epicenter? Depression, sleep disorders, sensory loss, so hearing, taste, smell, and heart disease. Those are at the epicenter virtually of so many non-communicable diseases. And that's what most of our prescriptions go toward, a high percentage prescriptions for those. Now, the contention in the book is that yes, there's been food changes, and that is certainly part of food technology, processed foods, changes in food. And I talk about going back to fermented foods as something beneficial, for example. Our ancestors used to be supported by those, but we've lost that over a couple generations, really, in large part. Although I was really glad to walk into a store here, and the first thing I saw was kombucha, and very, very helpful. So that's changed, but we've changed more. And it's estimated we've lost, if you compare us to, in the literature, to Amazonian indigenous populations, we've lost about 1/3 of our microbiome in diversity and richness. Now, they can be like us within a couple generations by moving to a suburb of Sao Paulo, and then by moving into Sao Paulo in the next generation. And they have basically no obesity, no diabetes in their natural setting, but within a couple generations they can have the same kind of prevalence of these as we have. So that's the ongoing, that's the reflection of what we've experienced since part of the 20th century. And again, none of it intentional. We had wonderful technology and we used it. We didn't realize risk, benefit, and unintended consequences. But now we know. So where does this affect us, and what are some of the things that we can consider? There are even microbes during the pregnancy. The placenta is now known to have microbes, and it is part of helping the immune system develop. But contrary to what I realized what I taught at Cornell back in the 70s when I first came there as an immunologist, about 50% of that's now viewed as wrong. That's how far back I go. Because I was taught that the human immune system at birth is good to go. You have everything you need, all the cells are there, and you're ready to go, and that's great. And what the book will argue, and with references, they're stuck at the back, and there's nothing interfering with the reading, so you can go there if you want, but they won't interrupt the readability of the book. But it turns out good to go instead is really good to self-destruct, because unless the immune system and the newborn baby co-matures with the microbes, it doesn't know what's safe and what's not safe. And eventually, it's going to react to peanut allergy, or to pollen, or to your thyroid gland autoimmunity. And that's exactly what happens. It's just a matter of what tissue, what stimulates it from the environment to cause a reaction, but it is not properly regulated. And so this whole thing about a regulated immune system in balance is absolutely critical. And the same thing happens for the brain. Neurodevelopment, we now know, requires and is a reflection of what's going on with gut microbes and microbes in other locations as well, and with the gut itself. So our physiological systems were never intended to be in isolation. We as humans spent much of the 20th century trying to be pure. We want to be pure. And I can tell you that we were never intended to be. And we're remarkably unhealthy. The purer you are, the less healthy you are. We are a walking coral reef. Now what that means is maybe our ego goes down a little bit, but we're incredibly awesome because of the thousands of species that we actually are. And the book will explain that by several different measures, you can argue that we're majority microbial. So by cells, there is some debate, and it references several sources in the book. The estimates range from around a small majority, around 60%, to a larger majority, around 90% of our cells are actually microbial. You can't see them, but what you can't see is your majority. But the genes, the comparison is even more in favor of the microbes. So the Human Genome Project, as you know, was going to solve human disease in large part. That was the goal. And a terrific effort went into it, and it is helpful. I'm not arguing it's not helpful, but it was underwhelming. In fact, fewer than half of the genes that were expected in number actually were found. So the total number of human genes is around 22 to 25,000, somewhere in there, but probably closer to 22,000. The earthworm has 20,000 genes by comparison, just so. Across humans, the estimate for the latest for microbial genes in humans is just under 10 million. So that means more than 99% of our genes are microbial. And they do things. They make cell surface molecules, they interact with the gut cells and the immune cells. They make regulators of our mammalian cells, small molecules that can regulate cell activity. They metabolize. And one of the greatest liver metabolism experts and toxicologists recently wrote 2 to 3 reviews on the microbiome and basically said, this is where the action is. They just, by numbers, their metabolic capacity for drugs, for food, for environmental chemicals is vast. And when they're not doing it, they're regulating what the liver does. They can exert an influence. Beyond that, there's a group working on neurodevelopment. And they've opened up a new area, John Cryan and his colleagues at University College Cork in Ireland and other research groups have basically shown that these things exert terrific control over our brain. And it's not shocking because it's been known that the gut makes more neurotransmitters than the brain. A lot of it by endocytes. But it turns out that there are particular bacterial species, Some of them make serotonin, some of them make dopamine, some of them make norepinephrine, GABA, acetylcholine. They make vast quantities, and they can regulate the endocyte, the mammalian gut cell production of neurotransmitters as well. So either through direct production or through regulation, they're exerting this massive control. And one of Krine's group's papers is titled Geppetto the Puppet Master, where he argues, yep, they're in control. And you can see this in terms of mood, depression, food cravings. And some of the experiments in animals, some of my colleagues have been able to take single bacterial species or the metabolites of those and show that they can take rats and mice, and if they put the bacteria in, they're very social. They play with their cage mates, just look great, normal, happy animals. And if you change that, so they're missing some of that, if you change the bacteria and the metabolites, they become OCD and they obsess on a ball, and they're completely oblivious to any other animal being in the cage. And they can flip that switch back and forth just by changing the bacterial composition of the gut. And the vagus nerve, there are like 3 different routes to actually control the brain. The vagus nerve is now known to be one of them. So there's a whole area called psychobiotics, where instead of taking heavy-duty medications for major depressive disorder, which is a pro-inflammatory condition, by the way, you're probably going to be able simply to change, take a bacterium, gut bacterium, probiotic, or metabolite, or change the balance there, and at least have that as an option instead of lifelong heavy-duty medications. And my contention in the book is that that's where pharmacology, that's where our new drugs are going to come from. They're going to have to work with and through the microbiome. And physicians are also not going to want to prescribe until they understand your microbiome profile. Now, why? Well, if they give you an antibiotic, and you may need it, you may have a very serious infection, it needs to be cleared. But almost all antibiotics, some to more an extent than others, kill microbes, kill bacteria that were not intended to be killed. So if a physician is killing a part of you that was not intended, the physician needs to put it back. That's basically what the book says, put it back. The paper where I won a Teratology Society award actually argued that missing the microbes, having an incomplete microbiome at birth, is equivalent to a birth defect. It's like not having an appendage or not having an organ that you need. And one of the reasons it's beneficial actually to look at it that way is it means it might be treated, because this is treatable. If it's actually recognized as a birth defect, they might do something about it. I want to mention, by the way, how I got into microbiome, because it is a fairly new area for most people, although bacteriologists will tell you they knew that back in the 1960s, and some did. They knew they were there, they just didn't know how important, or people weren't listening to them, is probably the way to put it. So I worked all my career on the developing immune system and protecting it, in terms of environmental toxins, drugs, food, ensuring it was as balanced as possible, and reducing non-communicable diseases. But the microbiome was new for me in 2012, and the way I got there was I was invited to write a paper for, of all things, a physics journal called Entropy, but it was an interesting question. The paper was to argue the one thing you could measure in a baby that would best predict a life course filled with health from one filled with disease. So think about that. What would you measure that would be the best thing to tell you the likelihood of a healthy life versus one with disease? And it doesn't mean that things don't happen. As we live our lives, things happen and they affect that. But if you could measure something there, what would it be? Well, I thought I had the answer. It surely was in my 30-some-odd years of research. That had to be the answer. And I started to write, and I got a couple of paragraphs in, and it was terrible. Absolutely terrible. I couldn't convince myself it was worth anything. And I was very frustrated. I went to bed that night, and I woke up at 3:30 in the morning from a dream. And I said, wow, I had just had this dream. Wow, do I have this idea? And the idea was what became known as the completed self hypothesis and was presented in the Microbirth documentary film. And it's the idea that a baby born without the microbes is an incomplete human. Never intended to be that way. And the extent to which that baby is then completed with a robust and diversified microbiome, not just for the gut, but the skin, the urogenital tract, the nose, the mouth, the airways, is the measure. How complete is the baby? The more complete, the healthier the life. And so that was really the simple thing. And it got seen, I thought, well, nobody will look for biology in a physics journal, but it was seen by the UK documentary filmmakers, and they featured it as one of the core themes within the movie, and that led to more lectures and more publications. And one of those lectures was seen by some book publishers, and that's why I'm here, literally. So it was waking up from a dream in the middle of the night that's really changed my career, in a sense, to recognize the missing piece to what I've been working on for my career. So what can we do? Well, the seeding of the microbiome at birth is critical. So that gets interrupted by cesarean delivery. And cesarean delivery can be medically necessary. It's risk-benefit, and it can be needed and should happen. But there also is elective cesarean, and that has been increasing dramatically in many, many parts of the world. And OBGYNs never, we simply didn't recognize the long-term impact. They were looking at it, looks like a healthy baby to me, short-term, and mother did fine, baby did fine, fairly safe procedure. But it's surgery too, so the mother got antibiotics. So that affects the microbiome that she donates. So I've now told OBGYNs that they have a new mission. It's wonderful. They can affect 2 lives. Because if you have a diabetic mother, you can modify the microbiome of that mother to help her health, and you're also changing and preparing the bolus, the microbiome that's going to be donated to the baby when the baby comes down the birth canal. One of the alternatives that's been developed, where cesarean's used by Dr. Gloria Dominguez-Bello at NYU, is a vaginal swab procedure that actually collects the vaginal contents that the baby would be exposed to coming down the birth canal, and takes the cesarean delivered baby and swabs in the mouth and around. And she's shown that while it's not 100% identical to vaginal delivery, it is very close. The baby's gut microbiome looks like the mom's that would be delivered. Now, what happens to the cesarean delivered babies? Well, it's not that they're microbe-free. Microbes will fill in. And where do they come from? They come from the hospital attendants or the birthing attendants, and if in a hospital, from the hospital itself. And those aren't necessarily the microbes you would choose for your baby. And the immune system then does not get to see a robust, diversified microbes. Its development is changed, and it's changed in a way that you're going to have inflammation problems. And one of the things we know about NCDs is you really almost have to have an underlying misregulated inflammation. Inflammation that is going on longer than it should, it doesn't resolve, it's not needed, it's not appropriate. And that supports non-communicable diseases. And there are some drug categories actually developed here through Harvard Med. Some of them are lipids that help resolve inflammation, and remarkably, they do things against seemingly disparate non-communicable diseases affecting different tissues. And it's like, wow, how does it affect this and this and this? And it's because you can't have those diseases if you've really controlled inflammation. Now, if you've already damaged your skin or your lung or your gut, if you've lost function, that's a problem. But inflammation is the tie that binds these together. Inflammation is needed for host defense, but it needs to end when it's no longer needed. So cesarean, but also the food changes. Now we know that things like food emulsifiers, a very recent finding, very common, polysorbate A carboxymethylcellulose, and the rodent information says that what they do is they kill 2 species of bacteria that maintain our mucus, our mucin layer in the gut. And that layer keeps some bacteria away from our mammalian cells and the immune cells, keeps them at a healthy distance. Good fences, good neighbors. And so when that's degraded, they come too close and they generate inflammation. And those are actually obesogens. Those will cause metabolic syndrome, obesity, diabetes in those mice. And we thought they were safe, but again, I'm a toxicologist, and what I'm here to tell you is toxicologists did their best on drug, chemical, food safety, but they were screening for mammalian human safety. The microbiome was never included. So guess what? Most of the majority of the existing drugs, where it's been reported, and food additives like these emulsifiers can be a problem. They can change the microbiome. So digoxin, the heart medication, works in some people, not in others, is toxic in some, and has a narrow range for safety. People haven't understood why. And it's because of one species of bacteria and the amount that you have in your gut. If you're out of balance, it won't work, or it'll kill you. And my contention is physicians are no longer going to be willing to just fly blind and prescribe these things if they have a way to measure the microbiome profile. And there are companies out there now with university consortia that are starting to do it. And there's more information we need. It's not that we're going to know more about the microbiome than we know now in the future. I mean, a lot more to be learned. But we know what's unhealthy, and we can do things about it that are beneficial. And that's the message of the book. And it really provides a resource list at the end, both for self-help and for physicians, and communicating with your physicians. And a lot of people so far on the tour have bought a copy for themselves and a copy for their doctor or their health professional, and to have that dialogue. So I do believe it's really going to change. I mean, the other thing is these are sitting at what toxicologists call the exposure routes. They're at our portals of entry. We see the world through our microbiome because they're on our skin. They're on the outside part of our gut, right? They are actually between us and the environment. And they're in the upper atmosphere surrounding the Earth as a bubble. And they're growing on the International Space Station. Although you can argue, did they get picked up en route? But they're very happy there. So you actually have gut bacteria in you that are related to gut bacteria at the bottom of the Marianas Trench, in the Dead Sea, in the Yellowstone Geyser, underneath glaciers in Antarctica, in the dark eating rocks. And they're in your gut. So one of the awesome things is you have kin in the most remote places on Earth. And it might be useful to recognize that. So it's no wonder that we feel better. People are healthier. You see articles. People are healthier when they're out in the environment. And we need to make our built environments so that they support the microbiome as well. So I think that I have definitely talked long enough, and I'm hoping it'll generate some discussion. I'd love to have questions, and thank you so much.
Back to the babies, because so 10% of babies are born by C-section. What is it? What are the other factors that are doing so that they are born with whatever damaged biosystem?
Right. So in some Brazilian hospitals, the statistic is 90% elective cesarean. Yeah. Okay. In England, it's like 20 to 30 or something. So, as you accumulate, so think about as you accumulate, yes, what you eat. So as you accumulate non-communicable diseases, so the mother may be obese, diabetic, with asthma. Those generally are associated with restricted diversity microbiomes. That's the donation under the best of circumstances, because we're a couple generations down the line on this epidemic already. So you have a mother who's being treated with medications for each one of those, but nobody's touched the microbiome. They've not tried to diversify it, they've not tried to modify it, very likely, and that's the donation. So even with vaginal delivery, you could have a restricted diversity microbiome delivered to that baby because the mother simply is carrying multiple NCDs.
I'm wondering if isn't part of it is the feeding practices.
Yes, yes.
I'm a retired pediatrician.
Absolutely. Yeah, so breast milk, let me just mention breast milk. So breast milk is a miracle thing. It delivers immune components the baby needs, right? It delivers some additional microbes that are part of the maturation. The microbe profile changes in the infant. So all the action is really within about the first couple of years that helps And actually, we have something called developmental origins of adult disease. There's this idea that you can get your physiological systems get programmed early. There are critical events and windows in which they happen, and you need the microbes there. And there are a lot of studies that are now showing that's the best. You can change things. My personal story of interrupting 30 plus years of over 100 rounds of antibiotics using a strategy involving rebiosis and dietary changes is in the book. But it's just one case, but it was obviously useful for me. So after continuous antibiotics over those 30 years, I'm now 2.5 years without needing any because I know what probiotics can help. So there is that. There is that we're donating this. We lost the 33% and we're donating restricted. And in breast milk, there are complex carbohydrates, and nutritionists and scientists have long wondered, why are they there? The mammalian cells in us cannot use them, can't digest them. They're indigestible. It's an indigestible part of breast milk. It's not there for the human mammal. It's there only for the microbes. Home for millennia for the microbes. So if you ever doubt the importance of the microbiome, if you think, oh, it's a fad, then just look at breast milk. Because nature does not waste, I assure you, nature does not waste resources like that. They're there to support the microbes. Yes.
Yeah. I mean, obvious listening to you, you're a true believer. But on a couple of things. In terms of cesarean sections and the practice that some people now have started to do with vaginal swabs and trying to transfer the microbiome, are they getting ahead of the data? Is there any data that supports that practice?
Yeah, well, there is. I mean, the NYU group has peer-reviewed publication on it at the moment. So there is some data. Will there be more data? Yeah. Is there a risk to consider? Strep B? Yes, there are risks, but this is all risk-benefit. So you look at some of the calculations on what's ahead in terms of obesity, type 1 diabetes, asthma, just as the first wave of diagnoses. That's a real problem, and you do have some developmental windows you've got to address. So one way or the other, you've got a risk of strep B to manage, or you've got decades of this sequence of diseases ahead, and you've got some choices to make. So it is reasoned risk-benefit decisions to be made, and that's between the patient and the physician. So I'm not preaching you've got to do it. I'm saying there are at least some options at the moment. Yes?
When a baby is delivered, how do you know that it has a defective microbiological profile? Yeah, I mean, they can do microbiome profiling, so they can do fecal samples, they can do skin samples, they can collect material, and they can do the genetic analysis on the composition. Is there a program to fix deficits or problems with the profile? There are strategies. I wouldn't say there's an integrated program. There certainly is not an integrated program to do it. But there are things that we know that are unhealthy in terms of these profiles that are generated and that are associated with obesity or associated with inflammatory bowel, or that either cause or hold those diseases states in place because of this regulation, and also with the neurodevelopment as well. And so there are changes that we know. There's actually thought to be about 6 sort of ethnic-related ideal types. And moving toward one of those ideal profile types should be a healthful alteration. Now, that destroys my retirement plan, because I had planned that I wanted to be the universal donor for fecal microbiota transplantation. I thought that would be. But it turns out that maybe moving to the healthier profile within your ancestors that have been eating certain foods and living in certain places for thousands of years is easier than moving to one half a world away. So for me to move, it's going to be European and African profile versus an Asian, and that may be easier. There is evidence to suggest that, because we now know that actually species boundaries can be determined by the microbiome immune interaction. That's a very recent finding from both mouse and from parasitic wasps,
So what happens is if you've got groups that have been geographically isolated and their microbiomes, immune systems honed separately, and then you try to mate them, it turns out that it's not just the chromosomes incompatibilities that will kill the hybrid or make it unfertile. The immune system will attack the microbiome and will kill the hybrid. And if you remove actually if you treat with antibiotics and remove the microbiome, the offending microbiome, the hybrid survives. So you can breach species barriers through the immune microbiome interaction that actually can help separate. So that suggests that compatibility is pretty important at a fundamental level. Yes?
When you talk about the microbiome, a couple things. Do you include viruses as part of the microbiome?
Absolutely. Fungi, parasite. Yes, absolutely. Absolutely.
And when you gave a figure of roughly 10 million genes in the microbiome, if we think of a bacterium as having, maybe 1,000 genes, something like that, then you're saying that we're talking about 10,000 species.
No, because they're strains. So within a species, and this is really important if you get into probiotics, by the way, it's not Lactobacillus acidophilus in this formulation is the same as in this, is the same as in this. Different strains of, regardless of what it is, will carry different combinations of genes. And some of those genes are going to be the ones you want metabolically to be helping you in a certain direction. So that's why you'll see in some cases on probiotics, you certainly see it in the research papers, they're identifying the strain. And that strain is important because it doesn't mean the same bacterial species will give you anywhere near the same result.
To the extent that there's a difference that may be significant from strain to strain, if one were to profile an individual's microbiome, one needs to profile, if you will, from 10,000 different strains and species what you've got.
Well, to be more precise, and so it is a level of precision, and there is again, as I said, there's much more information to be learned than we know. But for example, right now, obesity carries with it, and diabetes carries with it at a family level what's called a firmicutes to bacteroidetes ratio difference that is pretty easy to see and pretty easy to start to address. And that is, it's a little bit gross, but at one level it is telling you whether you're headed in a bad direction or headed in a good direction. And so there is more refinement. You're right, they don't have every gene, every species that any company is going to give you now, at least, but they can give you, basically every month there's more detailed information. There are changes going on that is in the right direction. We do know that standing pat, we know what will happen if we do nothing. That much we know. More drugs, more diseases, more disability, and that's not a very pretty picture. Yes?
Yeah, I'd like to know a little bit more about the science. You mapped out a causal chain going from messed up microbiome to the immune system to inflammation to NCDs. Could you give us an example, pick your favorite NCD, and trace it back? What happened? What happened initially to the microbiome?
Yeah. Well, there are changes, a lot of the changes involve innate immune cells and T regulatory cells, and there's several different populations, some of which they didn't know, 10 years ago. They didn't have the markers to detect them. Some called invariant natural killer T cells, for example, and there are several kinds of T regulatory cells and some that are pro-inflammatory or for different types of inflammation. The balance of those in the gut, calling for populations in the skin, there are signals from the microbes that will call in some of these cells in the skin to the underlying barrier, is really important in determining what's going to happen. So a lot of the action, as an immunologist, I was taught, the immune system, that's the thymus, the bone marrow, and the spleen, wherein the real action is not, yes, the development is there in part, but the real action is out at our barriers. So the gut has 70% of our immune cells. That's important. It means it's an important site. And so those regions in the tissue, microglia are brain macrophages. Their status has a lot to do with some of these neurodegenerative conditions. We want happy microglia. So the immune cells in the tissues and their status and their balance is exceedingly important in whether you're going to have things like asthma or inflammatory bowel. And that's exactly what's been seen in terms of colitis and a burst of one particular inflammatory cell population that one particular bacterium controls. And if it's there in the window of development to dampen, not actually to promote something, to dampen it down, then it's resistance to colitis. If it's not there, if it's absent, then in some genetic backgrounds, that's what's going to happen.
So what happens initially to mess things up, other than having a misfortune of being born by way of C?
So, I mean, if you go back and you look at germ-free animals and what happens then when they're challenged and the like, it's simply that the immune system there is postnatal education. The ratios of these cells, the balance of these cells has to be regulated, and it is regulated by co-maturing with the microbes in place. And in the absence of that, you're going to have misfiring. And that misfiring, we pay a price. It's usually oxidative damage and inflammation in some tissue or systemically, and eventually that's going to promote a pathology somewhere. So that's the problem, is this good to go at birth is just not, that was one of the 50% that really I don't think is true anymore. Ironically, in safety testing for the immune system, that some places that's probably still used.
The balance in there, sooner or later we'll be able to map it or get the profile. Like you said, we're developing in that direction.
Yeah, there's already directions would be useful to head in terms of what the literature says. And there's also vetting of at least some, not all. The probiotics are still a bit the wild, wild west. But there's vetting of some in the research literature that at least suggests these candidates have more information behind them, and then knowing what, much like TripAdvisor, knowing whether thousands of people actually saw any benefit or not is important.
You mentioned inflammation and, what's the other one you mentioned? But inflammation, there are things like have a baby aspirin every day, or Dr. Wild says that turmeric and ginger every day. And oxidation you mentioned, you can have CoQ10, but if you keep doing that, when that's controlled up?
So I mean, a range of antioxidants. The problem is you're trying to put a patch on a festering wound that's still there. They help. Antioxidants, a full spectrum of them, each one only does some things in scavenging and controlling, is useful. But if you're underlying, why not modulate the underlying immune system in the first place too? Yeah, it's a more holistic, comprehensive route, and that's what you can get at.
Can I go to my doctor and figure it out? I mean, if they take a swab or fecal matter, what?
Yeah, you can get analysis done, and I can tell you my doctor's older than I am. So part of the story is, he said lose weight. Well, I got part of the clue because I was at a meeting in Germany for 6 days that I helped organize, a residential meeting in Frankfurt, local food prepared in-house. No exercise. I mean, they're working as slave labor. They're working as 12 hours a day. And so I'm getting no exercise. I'm eating well. And all of a sudden, my pants don't fit. I've dropped a pant size because it was all inflammation. It felt normal to me, but it was all inflammation. So I said, okay, there's some foods back home in the good old U.S. that that is promoting ongoing inflammation, and I need to get at that. So I did, and I found out, eliminated them, and yet I can still get into one. I managed to get in. I was flying Southwest yesterday, and they had their computer system go down. I had no food, so of course I ran into what's available at the airport. That didn't go too well, and so I was looking for kombucha this morning. So I know, however, which probiotics in my own body. And I won't tell a group what I take because it doesn't mean they'll work for another single person. They're vetted in my own body. I know which ones will, within 24 to 48 hours, knock out an offending food that I happen to encounter. And some you get by accident. You just don't know they're in, a nice dish. For me, it will control it so that what was happening was the GERD went to recurrent sinusitis, antibiotics, in a couple weeks. And that would repeat 3 or 4 times a year. And Now I control it the minute I get the gut sensation that tells me I encountered something that was helping to promote the inflammation. Then I've got my own strategy for dealing with it. That combination of changing diet, yes, eat a healthy diet, but realize that your microbes are going to regulate your food cravings. So I ask people, I say, do you have a chocolate craving? Do you want a chocolate craving? Because there's probably a microbe, a bacteria for that. So I grew up in Texas in the South in the 1950s and 60s. So yes, Mexican food for sure, but fried chicken. If my microbiome uses fried chicken as an energy source, they are not going to like kale. No way. And that's actually what happens. That's part of the problem, I think, with just telling people go change your diet and lose weight, is they're fighting their own body, because the microbes have the neurotransmitters. They know how to control, whether you're happy about it or not, what you're craving, and they know what energy source they want. And you actually get resistance. So I believe that doing in concert a dual plan to change your diet in a healthy way, but also ensure you've got microbes installed that are going to use that as an energy source and is the best of both worlds, and will help more people be able to be more successful in that kind of effort. So again, my older than I am doctor said, I tell people to do it, they never do it. You did it, what'd you do? And then he wanted to read microbiome papers after that. Yeah.
I was wondering how exercise, we've been mentioning diet, how exercise, because as we know, exercise helps a whole bunch of things, from, even there's a recent study that it helps neurogenesis.
Yes.
How does that affect the microbiome?
In general, the literature says it's very good for the microbiome. I actually had to explain to some of the proofreaders one of the experiments, if you make mice run when they don't want to, and longer than they want to, which you might view as a form of torture, right? It damages the microbiome. On the other hand, if they're running of their own free will, and if we're exercising, I mean, there are experiments where they look at people before and after exercise, and it's beneficial. Yeah, and that's not shocking, but it is. I do want to say one thing. If we're talking about profiling and make it seem like it's really terrible and it's hard to do. There is a dog in the Netherlands, and there's a lot on dogs, and there's a lot on animals in the book too, not just humans, so I hope you'll enjoy that. But there's a dog named Cliff in the Netherlands who's been trained to detect the chemical signal, volatile organic compounds, off of Clostridium difficile, which is a very serious life-threatening infection, mainly associated with hospitals. He can detect it in individual patients and hospital outbreaks 3 days before any instrumentation or other technology. And think about how dogs greet each other. Is it face-to-face? So I contend they're actually asking, who are you microbially? How healthy are you? And might you be willing to share? In many cases. I used to yell at my dog, say, don't do that. And now I'm like, you guys are smarter than I thought. So no, they're incredibly, so if they can do that, they can detect other microbial signals too, not just C. diff. That's the point. So I actually talk about a breathalyzer in future doctors' offices. We've seen ultrasound move in. Why not get microbiome profiling before you prescribe? Keep in mind, NSAIDs, prolonged use of NSAIDs damage the microbiome. There are papers out there showing even naproxen and ibuprofen do it differently. So then you take proton pump inhibitors, but guess what? It damages a different part of the gut microbiome. They were never screened. Yes?
How long do you think until this view of the importance of the microbiome becomes mainstream medical practice? And I guess it's medical and agricultural, nutritional.
Yeah. So actually agriculture, one of the sections in the book talks, I was actually in, because I was hired to make healthier chickens, right? I was in the poultry department in the 80s when the salmonella outbreak happened. And it was both a bird farmer problem for their economy, and it was a human health problem because of contamination in poultry products. And you know what the solution was? It's called NERMI concept out of Finland, is they decided, you know what, we can load these birds up with Lactobacillus acidophilus and swamp out salmonella, and they did. And they showed that they could swamp it out. It wasn't vertically transferred through the oviduct to The chickens, the food products were safe. They basically dealt with the epidemic. We have 30 to 40 years of proof of concept data on that, and that's discussed in the book and referenced. And so they did that in an economic, where there's money on the line and in food safety. And there are challenges in getting some microbes to some parts of our bodies, and that's going to take some extra technology to be maximally effective. But that's the proof of concept in agriculture.
But it seems like the harder part is getting the doctors to change.
Yeah, well, I can tell you that there are some that are so on board that they're actually offering, are going to be, I think they're going to be certification type programs and in it. And so I think there will be increased training. I think as you see the pharmaceutical companies making drugs that work with the microbiome and through it and are relying on it, that's going to help as well. So keep in mind, cancer therapeutics now, they only work if you have the right microbiome in place. So they have to be metabolized, and it's in the microbiome. So cyclophosphamide and platinum salts, that's the case. So a lot of our drugs are relying on it. We can make existing drugs safer, reducing side effects, and make them work in more patients just by knowing the microbiome profile and, if need be, adjusting it before we prescribe. Yes?
How do you measure microbiome profile?
Well, it's done by genetic analysis on fecal samples, on skin samples, nasal swabs.
So it
again.
would be possible to take samples or somebody's nose and vagina, stomach.
Yeah, yeah. Or have a well-trained dog on hand, apparently. Yes, exactly, if you know what you're looking for. And there are some keystone species. So for example, you really want to know the status of those species that regulate the mucin layer. You do not want those damaged, and that's one of the ones that a signal for will be very important.
What disease I'm thinking about? We're talking about the mucin layer?
Well, I'm saying would be a priority. That would be a priority to go for. But there are, the profiles are very, very extensive that are generated. The moment they take a while, the question is moving that to a more rapid analysis type of system where you can at least ask specific questions for specific existing drugs. And it's not just drugs that are in this category. By the way, your actual personal susceptibility for arsenic toxicity is controlled by the microbiome. The extent of sulfur reducing bacteria determine how much of the worst metabolite of arsenic gets made. And so environmental chemicals, individual variation of risk for toxicants is influenced by it as well. So we just need to know, we need to know our microbiome profile eventually, and we need to manage it. It's like be the master of your garden is the analogy I use. So. Thank you.
The developed world tends to wear shoes, and they tend to wear shoes that are cushioned and certainly motion control. So our feet get weak and they get tight often.